today we're going to talk about, we're going to finish talking about T cell development. I think that will probably take the majority of the time today, and then I'll start with some information about trafficking, and the trafficking will then continue on Monday. And I feel like I always end up with my timing being a little weird around this part of these topics, so we'll make it work. Last time we looked at T cell development through some of the specific types of T cells that we saw here. So remember that our T cells, when they enter the thymus, they are double negatives, so CD4 negative, CD8 negative, and we can subdivide double negative cells into 4 populations of double negatives: DN1, DN2, DN3, and DN4, which are divided based on CD44 and CD25 expression. After being a DN4, the cells then become double positives. And we left our cells as double positives last time. So our double positives, of course, are undergoing testing, as we talked about, for their self-reactivity. And we have these different outcomes, 3 of which I mentioned on Wednesday. The outcome of no signal or very low signal, where the cells undergo death by neglect or fail to be positively selected, which is what happens to the majority of cells. They, of course, are not responding to MHC, so they're kind of useless. The ones that have very high affinity are negatively selective. They're actively killed, so we're deleting those cells. And only the ones that have kind of the low intermediate level of affinity are going to be positively selected and actually become the T cells that get to leave the thymus and go out into the periphery. We also discussed the fact that this is a little bit of a problematic scheme, because the only antigens that are available here to do this testing are self-antigens, which means that all of our T cells that leave the thymus had low affinity for self-antigen. So every T cell that leaves the thymus has low affinity, is sort of low affinity self-reactive. This is why we have a lot of checks on those T cells to make sure that they are dealt with peripheral tolerance mechanisms. We have lots of leashes on those T cells when they're in the periphery. But also, as I said, we're always kind of balancing right on the edge of autoimmunity with the way that T cell selection happens. And we'll get to this 4th outcome, I believe it's sometime next week, that I'll talk a little bit about. So this is kind of where we left things off. We also talked, I guess the other thing I'll say, just really briefly, is sort of the locations of what we've seen. So much of the early part of T cell development, especially with the sort of VDJ steps, are all happening in the cortex. Much of the selection parts, especially the negative selection, is happening in the medulla. And so I kind of think of a lot of the steps that we're seeing right now as happening in the medulla. Cells in the medulla are sometimes called medullary. And so we're going to see a couple of the structural cells of the medulla on an upcoming slide. So if I talk about the medullary epithelial cells, I mean the epithelial cells in the medulla, the structural cells in the medulla there. When we think about T cell selection, as I've already kind of told you, there's sort of this issue that we have to worry about where all of the cells are testing against self-antigen and are leaving if they have low levels of self-reactivity. And that sort of is this issue that means we need peripheral tolerance. When we talked about B cell development, there was also an issue that came up with B cell development that made us realize peripheral tolerance was going to be important. And so what was our B cell problem? Yeah, Maddie.
B cells only react to antigens.
Yeah, so B cells only react to antigens in the bone marrow. They don't get a chance to test against other antigens. So, insulin or proteins in your eye or any other tissue-specific protein is not present in the bone marrow, so we can't delete B cells that are self-reactive for those. And so you might look at this model of thymic selection and kind of wonder about the same issue in the thymus of how do we deal with tissue-specific antigens? How do we deal with T cells that respond to tissue-specific antigens? Because we already got a problem with T cells. It would be bad if we had two problems. That would seem not cool. And again, when I was an undergraduate, the answer was, we deal with it. Early in the time that I was in graduate school, the mechanism for this was also kind of worked out and was described. And so what we now know is that the thymus actually transcribes and translates every protein in the genome. So every protein is expressed in the thymus. So for example, you might have some proteins that are just in the retina, some proteins that are just in the ovaries. It turns out that just in the retina means retina and thymus. And just in the ovaries means ovaries and thymus. And so you actually make every single protein in the thymus. So there is no protein that's not in the thymus. And so we have this protein that helps us to turn on all of our tissue-specific proteins in the thymus so we can actually delete all of those self-reactive proteins. The cells that are doing this are the thymic epithelial cells, specifically the medullary epithelial cells in the thymus. So the medullary epithelial cells in the thymus make every single protein so that T cells get a chance to test against every single protein. Medullary epithelial cells are also unique because they express MHC class II. They are sort of the important for us cell type that is not one of the professional antigen presenting cells that still presents on class II. And this means that the cells can actually test themselves against class II, not just class I. Remember, every single nucleated cell makes class I, even DCs, macrophages, and B cells. It's just that DCs, macrophages, and B cells also make class II and present on class II. Thymic epithelial cells are the same way. They also present on class II, and that's really important so that they can help T cells see every possible antigen for development. And so those thymic epithelial cells, those medullary epithelial cells, are making every single protein in the thymus. This is done as a result of a protein known as AIR. AIR stands for autoimmune regulator. This textbook phrases AIR really nicely. It says, under the control of the AIR proteins, thymic medullary cells express these tissue-specific proteins. If you study AIR, and you study kind of different aspects of AIR, almost everything that you study about it makes you say AIR is a transcription factor. It's a transcription factor that leads to the transcription of all these different proteins. Except if you do an assay to try to make it actually be a transcription factor, it won't be a transcription factor. And so how AIR controls things, we still don't totally understand. It in every way seems to be a transcription factor, except that it doesn't do transcription in a test tube. So it's somehow under the control of AIR, but exactly how AIR does that controlling, not totally sure. This was first described actually in two different sort of sets of experiments in both mice that were missing AIR and in some patients. And so this is some data looking at some of the genes that get transcribed in the thymus in the absence of air. Actually, these aren't patients, these are in the mice. And so these are some of the genes whose transcription seemed to be really messed up in the mouse thymus when air was gone. And you can see that many of them are in red, which means that normally those are things we think of as being in one specific tissue. So your thymus normally makes casein, which is in mammary tissue. Your thymus normally makes trefoil factor, which is in your GI tract. It normally makes major urinary protein, normally makes a salivary protein. It normally makes oxytocin. It normally makes preproinsulin. It also makes things like insulin-like growth factors, cytochrome P450, other things that are known to be in sort of just very separate special tissues. And so you can see all of these genes are controlled by air. So many of these tissue-specific genes are controlled by air. As I mentioned, one of the things that was involved in the discovery of air when this all happened was some discoveries in some patients that had mutations in air. So based on what I've told you about air right now, what would you imagine happens to a patient if they have a mutation in air? Yep, Sebastian. Okay, so we don't delete cells that would respond to tissue-specific antigens. And so they're going to have these T cells that are going to get out into the periphery, into the rest of the body, that can respond to tissue-specific antigens. And so what probably happens to those patients? Yeah. They have some sort of autoimmune disorder. But I want to think about that for a second. What might be sort of unique about the autoimmune disorder or the autoimmune issues in these patients? Yeah, Justin. So Justin wants to know if that's specific to one organ. What organ do you think it would be specific to, Justin? So that's how we usually think about autoimmunity, is usually we think about the fact that there is a mutation that sort of impacts the retina, so you have autoimmune disease in the retina. But remember, air is turning on transcription of all the things in the whole body. So Jamie.
So they would have a widespread, like, global body autoimmune.
So probably they're actually going to have multiple autoimmune diseases against multiple parts of their body, all at the same time. Because it's not one T cell that sort of slips through the cracks here. It's all the T cells that should have been controlled by air. Now, yes, some of them are going to get hit by peripheral tolerance, but there's a lot of T cells here that are potentially a problem. And so these patients are going to have a bunch of autoimmune diseases all at once. And in fact, every patient is probably going to have kind of a different combo of some autoimmune diseases. The disease that we see in patients who don't have air or who have a mutation in air is called APESED. I'm going to tell you what APESED stands for. You do not have to write it down or be able to remember it. APESED stands for autoimmune polyendocrinopathy with candidiasis and ectodermal dystrophy. Again, that's what. You don't have to write that down or remember that. And so the idea is these patients have multiple autoimmune diseases, so autoimmune. Many of them impact the endocrine system and endocrine glands, and so polyendocrinopathy. Weirdly enough, these patients also get recurrent fungal infections, so candidiasis, and there seem to be some other cells that are off, and so that's ectodermal dystrophy. And so here you can see a cohort of patients with APESED. And you can see what % of the patients have each of these different types of autoimmune symptoms. So you can see that 85% have hypoparathyroidism, 72% have adrenal failure, 60% of the women have ovarian failure, 11% have diabetes, 14% of the men have testicular atrophy. Also, 100% have these yeast infections, candidiasis. A lot of them have weird things going on with their fingernails, this nail dystrophy, and on and on and on. And so each patient has a different combo of these. So it's not like there's a standard set of symptoms. And this is due to some group of T cells all getting out of the thymus that shouldn't have. Yep, Jamie.
I have two questions.
Okay.
Did you say this was from a lack of air or just a mutation?
It's loss of function of air. So it's a mutation that makes air not work, whether that's a lack of or, yeah.
And then how do they go about diagnosing this?
So a lot of it is looking at, so it's actually, they do the nail dystrophy and the candidiasis, as well as just sort of general autoimmune issues. But you can imagine it is a tough diagnosis. And generally, the patients are probably going to present with one thing, and then later they're going to present with another thing. And it's probably not the first diagnosis these patients get. So, weirdly enough, in the mice, the biggest problem the mice have is the mice have this crazy dramatic autoimmune destruction of their salivary glands. So if for some reason in mice, this particularly means their salivary glands get destroyed. And what the deal is with that is sort of less clear again. And so this is kind of what an autoimmune disease looks like when it is a failure of central tolerance. They look a little different when they're a failure of peripheral tolerance. And air is acting largely in these medullary epithelial cells that are making all sorts of different proteins to allow for the deletion of those cells. And so this is sort of the overall description of what's going on with T cell development in terms of locations. So we've been kind of focusing on those double positives. When a cell sort of gets to that point of making TCR alpha and beta and getting a good signal, it will eventually become a mature T cell and get ready to leave the thymus. When that cell is on its way out of the thymus, it then goes to being either a single positive CD4 or a single positive CD8. So once the cell has kind of dealt with all the steps we've seen, it will become single positive CD4 or CD8. And those are basically the cells that are on their way out. And it will leave the thymus. And now this cell is ready to get some signals through its T cell receptor to proliferate, survive, and do a thing against microbes. And so you can see these mature T cells here. Basically our cell as double positive will have CD4 and CD8. CD4 and CD8. If it gets too high of an affinity signal, it'll be negatively selected. If it gets that sort of medium signal, it'll be positively selected and become the mature T cell with either CD4 or CD8. It's a single positive, where it can start to do its thing when it gets a signal. One question that is in the field is how the cell decides to be a CD4 or a CD8. How does it know, I'm going to stop making CD8 now, only make CD4 and become a CD4 positive T cell. Or I'm going to stop making CD4, I'm going to only make CD8, and I'm going to become a CD8 T cell. How does it make the choice which of these two directions to go down? That is also still debated. There are a couple of different models. And so just to show you sort of some of the different ideas of how this really works, I'm not going to talk about this model that's at the bottom. I'm only going to talk about the two that's at the top. One idea about what happens is that the cells actually are getting signals and figure out, do they get a better signal with class 2 and CD4 or class 1 and CD8? Whichever one gives them a better signal, they pick that one and get rid of the other one. The alternative model is that the cells just pick one. They choose, and then they see if their receptor still works. And if it doesn't work, they die, and if it works, they live. And it's actually a little bit hard to tell the difference between those models in a number of different experimental settings. And so there's still a fair amount of debate about that. So know that that piece is less clear. However it happens, the cell will then leave the thymus and become a mature cell that is able to go into the periphery. So if you recall, this is sort of that flow cytometry plot we see on T cells just straight out of the thymus. So we just take cells, we mush them up like you did in the lab. We run them straight, we've seen them with CD4 antibody, CD8 antibody. We run them through the flow cytometer. This is what we get. Or more appropriately, this is what it really looks like. My grad school class, sort of as a joke, decided that the thymus bird was our mascot. So that's kind of what it really looks like. This is kind of how we depict it. And that's what we see in the thymus. We can also then look at the T cells that come out into the periphery. When we look at organs of the periphery, the two organs we most frequently look at will be the lymph node or the spleen. We see something that looks a little bit different. So if we just take our spleen, like you did in the lab, and smush it up, and run it, stain it with antibodies against CD4 and CD8, there's no reason why one has to be on the X and Y. I just figured I'd be consistent from this graph to this graph. So I actually just do that experiment. I'm going to see this. So I'm going to see 3 populations. You can see that I draw them as being pretty similar in size. And I really don't see much, if anything, in this double positive. You should see very few, if any, double positives. This is real data from a paper. But typically when we think about this or when we draw this, this is what we draw in the periphery as for either a lymph node or from a spleen. Because those double positive cells are cells that are only made during development of T cells in the thymus. They don't leave the thymus. And so we won't find them anywhere other than the thymus. So if I look at a CD4 versus CD8 plot, I can tell right away if it's thymus or if it's lymph node or spleen. There's one other way that sometimes we see our lymph node or spleen plots depicted. And so I want to kind of try to draw this out. Again, this is going to be subject to my excellent art skills. So you could take your spleen, like you did in the lab, and you could do flow cytometry. And so one thing you could do, is it that way, 8 and 4?
Yeah.
Okay. And you get this. So if I do my cytometry this way, what are these cells? Yeah, these are the CD8 positive T cells. What are these cells? cells? Sorry. What you say, and then whoever, Krishna, were you the other one? Okay. So what do you say, Jamie?
I said they're lacking both.
Okay, Krishna, what were you going to say?
I said double negative.
Okay. Yeah, Sebastian. Yeah, in fact, those cells are the non-T cells. They may also be some of the gamma deltas, but really these are the not T cells. The cells that come out of the thymus are all the CD4s or CD8s. Really, those other cells are non-T cells. So if you decided you wanted to be really particular, you could look at CD3 versus something else. It actually does not matter to us what the something else is. Out of the spleen. And we're going to see some cells that are CD3 positive. Whether there's something else positive or negative kind of depends on what the something else is. If you're CD3 positive, you must be a T cell. Remember that CD3 is that co-receptor that works with just the T cell receptor. So only T cells have CD3 on them. I could now take just those cells and do more cytometry on them. This is called gating on the CD3 positives. So I could gate on the CD3 positive cells. And when I do that, I will get this plot from my spleen. I would get the same thing from my lymph node. So what you can notice is the difference is that one of the populations is missing. Now we have our CD8 positive T cells, and we have our CD4 positive T cells, but we are missing the not T cells. Because we did this step earlier to find the T cells and only analyze T cells. So we gated on CD3, so we could only see the T cells, and so we get rid of those cells. We don't ever usually talk about the gating with the thymus, and really it's because all of the cells in the thymus are T cells, so we don't worry about gating. But when we go into the periphery, the spleen has B cells, the spleen has macrophages, the spleen has NK cells, the spleen has all the kinds of cells. And so we might have to gate in order to get a totally clean T cell prep. So this is what we would see. These are kind of some of the differences you might see in some of these cell types. Much of what we have learned, and much of the way that we have learned about T cell development is to look at mice that have different kinds of fancy genetics and then checking to see what happens. And sometimes we look at a plot of the thymus to kind of see which of those populations are present. Sometimes we look at the plots from the periphery and see who makes it out of the thymus. And so I've got a few slides now that are about some of the sort of specific experiments we have done or people have done to help us understand T cell development. And so I want to show you some of those experiments and kind of what the outcomes are and tell you kind of which piece of this whole T cell development process we've just talked about was learned by that experiment. And then we'll move to something other than T cell development. But first we're going to go through these experiments. One of them I know confuses the heck out of people every year. So we'll go slow on that one and make sure you let me know if you have questions on that one. So one of the sort of easier things we can do is we can use mice that have a knockout either in MHC class 1 or in MHC class 2. These are actually pretty hard knockouts to do because you have many class 1 genes and many class 2 genes. I would say that if you are trying to answer a question on a problem set or an exam, you should probably not say like, I'm gonna knock out class 1, because really that means you're knocking out like 6 genes in a mouse. Or you're not going to say, I would have a knockout of class 2, because that's, again, a knockout of a lot of genes. So know that this is actually not really one knockout. Just be aware of that. But what we can do is, if we have control mice and we look in their thymus, we see double negatives, we see double positives, we see CD4 single positives, and we see CD8 single positives. If we get rid of class 1, we see double negatives, we see double positives, and we see CD4s, but we don't see CD8s. One of the things this tells us is that double negatives and double positives must have come first. Those must have been earlier steps than the CD4 and CD8s. These must have been a late step. Similarly, we can have class 2 deficient mice. We have double negatives, we have double positives. We don't make our single positive CD4s. We do make our single positive CD8s. So again, this tells us double negatives and double positives must be earlier steps than the single positives. It also tells us that MHC is in fact involved in T cell development, which, yeah, it is. And so we can sort of see that from some of these experiments. We can also do some other types of experiments where we can look at the T cell responses in our mice. If you recall, when we talked about B cell development, we talked about some genetically modified mice that were used in order to help us understand B cell development. Those mice were called BCR transgenic mice. In the BCR transgenic mice, we added 2 new genes to the mouse's genome. One of those genes was a pre-rearranged heavy chain. One of those genes was a pre-rearranged light chain. Then, when every B cell tried to develop in the bone marrow, they would already have a pre-rearranged heavy chain that worked. So why rearrange more? We would have a Lollick exclusion. We would already have a pre-made light chain. Why rearrange more? So we would have a Lollick exclusion. And 100% of the B cells in those mice would become B cells responding to our antigen of interest. We saw those mice in those kind of experiments with B cell development. And one of the reasons why those were super important is that people argued about B cell development and deletion and what was going on. And it was really hard to track the one B cell that was specific for your favorite antigen. And some people are like, no, it didn't get deleted. You just couldn't find it because it's hard to find a needle in a haystack. And so they had to come up with this type of experiment to let them make the rare cells not rare anymore. We can make similar types of mice to help us study T cells. And so instead of being B cell receptor transgenic mice, these are T cell receptor transgenic mice. When we do this, we start with a T cell. Here's a T cell. This T cell that we picked, that we decided to start with, was a CD8 positive T cell. In this case, it could be a CD4 or a CD8, but in our example, we picked a CD8. And we picked a CD8 that recognizes the influenza antigen on a class 1. I believe it was on its K of K, but I don't exactly remember right now. So we actually stole this from a mouse. Basically, we gave a mouse influenza. We said, make some T cells in response to influenza. We found the perfect T cell. It was red. It was so perfect. And then we did some PCR, and we got the alpha chain and the beta chain that had rearranged in this T cell. So we got them out, and then we took those genes and we put them into other mouse embryos. So now the mouse genome has the pre-rearranged alpha and pre-rearranged beta chain from this T cell that I picked because I liked it so much. When I do that and I let those mouse embryos develop, 100% of the mouse's T cells are clones of this T cell. 100% of the mouse's T cells are CD8 T cells with this alpha and beta chain that recognize influenza antigen on this type of MHC. That's because 100% of the T cells are going to make this alpha and this beta chain. And when those T cells, which had the transgenic receptor recognizing class 1, started developing in the thymus, they were double positives. They tried to test, and the only ones that actually worked were ones that had CD8 and could grow up to become this kind of T cell. And in fact, if I look at the thymus of this mouse, I'll see some double negatives, I'll see some double positives, and I'll see CD8s, but I'll see no single positive CD4s because none of them develop. If I look in the periphery of this mouse, I'm gating on CD3. 100% of my T cells are CD8 T cells. And in fact, they all recognize influenza. So now I got like an infinite source of my favorite T cell. Yes, this mouse would get very sick if it escaped my mouse facility and like went into the world and got infected with something other than influenza. It would be out of luck. But I can use it as a source of these cells really easily, and I can track the development of my cell of interest in this mouse really easily. If I put in a transgenic receptor that recognizes an antigen presented on class 1, I will only get CD8 T cells, which is what I just showed you. If I put in a transgenic receptor that recognized an antigen presented on MHC class 2, I would only get CD4s. But you could also think a little bit about the MHC type here. If I put in a transgenic receptor that is recognizing an antigen that's presented by the MHC of type A, so one genetic type of MHC, type A, then the single positive cells are going to mature. And if it was a class 1, they'd be CD8s. It would work. Hooray, good job, us. If, however, I put in genes for a receptor that was restricted to MHC type A, and I used a thymus that was MHC type B, then all of those T cells are going to develop. They're all going to be double positives. They're all going to try to interact with MHC in the thymus, and none of them are going to interact. And they're all going to be sad because they get no signal, they get no pat on the head, and they're all going to die by neglect. None of them can get a signal from that thymus. And so in the end, we're not going to get any single positive T cells out. And this can help us understand T cell development. There was a famous experiment that used this as one part of it. So you're going to see basically this same phenomenon in the experiment that I show you. There's also one other piece to that experiment. And so this is, again, that same idea. It's even another experiment. So I start with a mouse of the H2K haplotype. I pick one of its T cells. It happens to be the CD8 that recognizes influenza. If I put those genes in a mouse of the same genetic background, I get double positives. I get CD8s. It works. If, however, I put those genes into a mouse that is otherwise of the H2D genetic background, I get double positives. They try really hard to develop, and they are looking for MHCs that they can match so that they can get a signal, but they get nothing. And they all die. And so there end up being no CD8s in this mouse. The actual experiment was done as is shown here. It's really the same experiment that I just showed you in a lot of ways. The difference is that the antigen is not an influenza peptide. The antigen is a peptide called HY. HY is specifically presented by H2D. Is that class 1 or class 2? Is H2D a class 1 or a class 2? Class 1. And it's the specific kind of H2D called D of B. Okay? So what kind of T cell is this? Is it a CD4 or a CD8? Since this is the class 1. CD8. In fact, always remember, 8 times 1 equals 8. 4 times 2 also equals 8. It's a good way to remember. It always has to equal 8. So they found this CD8 positive T cell that recognized this antigen, HY. And they got the alpha and beta T cell receptors from this mouse. And then they put those into mice of either the right genetic background, a B genetic background, or a D genetic background, the wrong type of mice. Okay? So it's exactly the same experiment I showed you before. And this is the outcome. I'm also going to draw the outcome in plots. I'm also going to show you the outcome in terms of the data from this experiment. One thing I will tell you is that the data is messy in the real data from this experiment. It's because when they did this experiment, it was 1992, I believe, they did not know that alpha did not allelically exclude perfectly. And so they got some cells that did weird things because of alpha. And so if you look at the data and you're like, there's some stuff on here I don't understand, Dr. Barker, the answer is because the data is messy. Okay? So remember, if we look at the thymus from our normal mouse at the mouse store, this is what we see. We see some double negatives, we see double positives, we see single positives. So that's this. Now we can look at the thymus of our mice. We can look at the thymus. We can look at the thymus from our mouse that has the matching MHC type, H2D, or the mouse that has the not matching MHC type, H2B. And so these are the data, and basically this is what they see. So if they look at the mouse, oh, actually, hold on. B is the match. D is not the match. There we go, that's better. They look at the mouse where they have the matching MHC type. They see some double negatives. They see some double positives. They see some CD8s. We'll pretend they don't have, that's not there. This is exactly what they were hoping to see. We put in a receptor that bound to a class 1 presented peptide. That makes us CD8 T cell. So we got 100% of our T cells were CD8s. 100% of our single positives were CD8s. Got no CD4 single positives. That's what we wanted. When we actually did the mixed genetic background, where the mouse had at least some of the right genetics, we got the same thing. But then, when they did the experiment where the cells had the wrong genetic background, they got some double negatives, and they got some double positives, and they got no single positives. Again, because these cells are double negatives, they go through the process of VDJ recombination. They're not self-reactive, so we don't have to delete them. It's not like imperative that we kill them right away. But those cells sit around and are like, hello, give me signal. Let me keep developing. And they get no signal, and they die. So they never go any further. And so that kind of does tell us that these must be the last steps. These must be the first 2 steps in some order. And they tell us that MHC and the antigen are important. But what this experiment doesn't tell us is what happens if the cell was self-reactive? What if that antigen was actually an antigen that was a self-antigen? But actually, it's a tricky experiment because we can figure that out too. So as I told you, our antigen in this case is called HY. HY is a protein that is encoded on the Y chromosome. That means HY is a foreign protein in females and is a self protein in males. And so first they did this whole experiment in females, where this was a foreign antigen. For all intents and purposes, it was influenza, as far as they cared. It was some foreign protein that's not part of their body. So their thymus did not ever have this protein. This was not a self protein. But then they repeated the experiment and looked in the male mice, where now this antigen is a self antigen. And the thing is, we know the T cells can develop normally if they're not self. We know it works if they're not self. So then what happens if they're self? So now they also look in the male H2B mouse, where the antigen is a self antigen. Sometimes people are like, what about the male H2D? And then it's just like, it's not matching. Again, really. Like, that one doesn't matter. The male that matters is the H2B, because that's a self antigen case. And so they did the exact same experiment in the male H2B mouse, who in fact expresses HY as a self-antigen, whereas the females did not express HY. It was a non-self antigen. And when they did that, they saw double negatives, and they saw teensy bit of double positives. And that's it. There's the very hideous data. That's a better way of saying what they saw. If you do the experiment the right way, we now know how to do it. That's what you see. So what happened to these cells? They're double negatives. They start trying. They become a double positive. What happens as soon as they're double positives? What is it, Jamie? They get to, as soon as they're double positives, like, oh, no, bad. We gotta kill you. It's not like they had to wait around and just wither on the vine. We actively killed them. And so we see basically no double negatives. We have a few that got into the process and tried to start, and then they immediately get killed. And really the only cells we see are the double negatives. And when we do this experiment, that actually tells us, so remember, we knew that these were step 3. But we didn't know, is it 1 and then 2? Or is it 1 and then 2? We didn't know. They didn't know. What can we tell now that we've done this experiment? Yeah, Jamie. Yeah, so we can tell that double negative must be first. That must be the first thing the cell tries to do. The next thing the cell tries to do is double positive. In this case, it gets killed in that process, and we can't get to the later ones. Here, it gets allowed to make double positives, but the single positives must be last, because it gets killed before they get to do those. And so these experiments actually really the experiments that explained all of T cell development. And so this is how we understand all of this information about T cell development. Yep, Jamie.
On the chart on the left side,
Yep. It looks as though some of these cells, so 2 reasons. Part of it is that some of the cells are spontaneously upregulating CD4. The problem with this whole experiment, the problem with all along, is that TCR alpha does not allelically exclude 100%. So some of these cells tried to rearrange and make another TCR alpha, and it seems like that primarily gave them CD4 cells. When we do this experiment now, you not only do the genetics that are shown here, you also have this mouse be RAG deficient. So no more recombination can happen at all. So it's not possible for any like weirdo recombinations to happen because you have no RAG. And so now we do it in RAG deficient and we get clean plots that look like that. Your textbook, as it should, shows the real data from the real experiment, which is messy because they didn't know that they needed to use RAG deficient at that point. So that's how we've learned, in fact, that's where all of those tables in your textbook came from, is from those types of experiments. And so now we have come to an interesting part of the course. At this point, we have now seen the development of all the types of cells I'm going to tell you about. We now are ready to look at the periphery. And we now are in the periphery. We have finished bone marrow. We finished thymus. We're now finally in the periphery. So we're switching gears now to see that we're actually going to get to watch our cells do something against a microbe. We've made them. We've built the whole immune system. Now we get to use it. So we're looking at our T cells in the periphery, or more specifically, we are now going to be looking at adaptive immune responses in the periphery. So we've got B cells that come out of the bone marrow. We've got T cells that come out of the thymus. And they both are going to end up in blood flow and sort of be going around the body in the blood, going through the heart into the blood. And then we need to kind of think a little bit about, okay, so where do they go? Where are these events happening? How does all this work? We're calling this events in the periphery. Where exactly are the cells? Is the periphery Mars? Where is the periphery? So you can see in this figure a general idea of where those cells are going. You can see that this shows about 10% of them going into barrier organs. I will tell you that our estimate on that number is weak, to say the least. Because in order to actually do this, like if I wanted to know right now, what percent of your T cells are in your skin? The way I would find that out is I would take all of your skin and put it in a tube and mash it up and extract the T cells and count them. And I don't know about you, but I am not volunteering for that experiment anytime soon. Similarly, if I actually really want to know how many T cells are in your GI tract or in your intestine or in the wall of your intestine, I would actually have to take your intestine and like go through it and find the T cells. So our numbers here are really, really, really estimates. I honestly have seen the numbers for the intestines vary widely. If you see different numbers than what are shown here, just know that there's a lot of sort of experimental issue here. You can also see that some fraction of the cells are going to go into other secondary lymphoid organs. Some of them are going to spend a lot of time in those secondary lymphoid organs. Some of them are more going to spend their time in the blood. You can also see some of this info here. If your cell just stays in the blood, it makes a complete circuit of your body, heart, all the way around back to the heart in 30 minutes. So every 30 minutes, it makes a circuit. It could also leave, and different percentages of them will leave and go into some different organs. And the key thing that's going to happen in many of those different organs, we are largely going to be talking about the lymph node, is that those cells are going to look for antigen. You could imagine it would be really hard if that cell had to go to every cell of your body. Do you have my virus? 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 Take a long time. The virus would then have a lot of time to reproduce before the cell found it. And so instead, basically the cells just go and check the secondary lymphoid organs. And so pretty much everything we're talking about when we talk about immune responses in the periphery, at least to start, is really lymph nodes or spleen. It's really a secondary lymphoid organ where we're seeing these processes happen. Those secondary lymphoid organs do have really specific structures within them. So you will see that they all have blood vessels in as well as blood vessels out. They also have lymphatic vessels in, which are the afferent lymph vessels, as well as lymphatic vessels out. And the cells can choose to circuit the body in either the blood or in the lymph. There are also different areas, particularly some areas that are known as B cell zones and some that are T cell zones. I bet you can imagine what lives in each of those zones. And we also have a lot of specialized vessels, which I will say more about next time. And those specialized vessels are referred to as high endothelial venules. They are specialized to let lymphocytes leave the blood and go into the lymph node there. And for the longest time, I just knew it's a high endothelial venule, HEV. Sure, great. I didn't actually know what that meant for a long time. What high endothelial venule means is that the cells of them, the endothelial cells, are high. Another way to think about this, the idea that the cells are high is that they're tall and skinny. That's what it means that they're high, is that they're tall. So we have a whole bunch of really tall, skinny cells making up this wall of this vessel. And all of this is incredibly important for our adaptive immune response. So if you remember, when we talked about the adaptive immune response, we see that the adaptive immune response really peaks, a week or so after infection. These are data From my PhD, I infect on day 0. You can see that I start to see a good CD8 response at about day 10. I really get my peak at day 17. And so really what we're going to be thinking about, honestly, for all of next week, maybe in sort of the next couple weeks, is what the heck takes so long? Why do the cells take all this time? What are all the things that have to happen in order to make this good adaptive response? And some of those steps are shown here at the bottom. They're also shown on the next slide. And so, yeah, we have to have infection, but we've got to do something where we can move the antigen. So we've got to move the antigen to get to the lymph node. We also have to move the T cells and the B cells to the lymph node. So part of this is move the antigen. Part of this is move the T cells. Then the B and T cells actually have to engage with their receptor, and they have to do some cell division and some differentiation. So they actually will change their function while they're in the secondary lymphoid organ. And then we finally have to have those cells travel again to wherever the infection was. In the case of, say, SARS-CoV-2, you'd have the antigen travel from the lung to the lymph node. The T cells would have to go to the lymph node. The T cells and the antigen would hang out in the lymph node. The T cells would get activated, would divide, would differentiate, and then the T cell would have to go back to the lung. And only then could the T cell actually do anything about your SARS-CoV-2. And so all of those events are the events that we are going to be seeing over the next few class periods. You can see that here as well. This is kind of showing us the timing in terms of both B cell and T cell responses. So you can see that it only takes a few hours for the antigen-presenting cells to sort of get there, up to a few hours for the antigen to get into the lymph node, but then at least a day for the T cell and the antigen-presenting cell to start interacting. And then the T cell will start changing over the next 4 days or so. And then we still have to have more traveling. The B cell, you can kind of see again, sort of that we have a few days for all of these events to happen. And we're going to see all of these events in specific detail over the next few class periods. The first event that we have to think about is the traveling event. How the cells travel to the lymph node. How they know how to get to the lymph node and not, I don't know, to your nose or some other random place. And that's where we will start on Monday, is the specifics of this trafficking pathway.